Hi, I'm Eva Prakash. I'm an undergraduate student at Stanford University working at the Kondaje Lab, and I'll be presenting our poster on towards more realistic simulated data sets for benchmarking deep learning models and regulatory genomics. As a brief overview, a common goal in the field is to build classification models that learn to distinguish a positive set of DNA regions associated with a specific function from some background or negative set of DNA regions. An example of positive regions might be those that are accessible in a particular cell type, and negative regions might be regions accessible in other cell types. Complex models such as deep learning models or support vector machines often achieve state-of-the-art performance at these tasks and are then interpreted for novel biological insights into what they learned. In order to evaluate the quality of the interpretations of such models, there are two main approaches that have been used. The first is to create a completely simulated data set by sampling motifs from a position frequency matrix and implant them uniformly into a randomly generated background sequence. In this approach, we have the benefit of being in control of the exact ground truth because we are deciding the motifs and inserting them into known positions in the sequence. However, the problem with this is that these simulated sequences may lack the complexity of real biological data. Real protein binding is highly cooperative, with many factors often co-binding and affecting each other, and we also don't capture any positional preferences in motif occurrence. Alternatively, we can use a real data set based on real genomic sequences. We identify relevant motifs in a cell type based on prior knowledge and search for them in each sequence. The obvious pro of this is that it's real representative data. However, it is difficult to ensure we aren't missing any motifs when we don't definitively know the ground truth. Our solution is an approach that's a hybrid of real data and complete simulation. For our positive set, we ran the Homer Motif Discovery tool to identify the exact positions of all known motifs in real genomic sequence data. We used FIMO to record exact positive motif locations in both positive and negative real sequences. We then dinucleotide shuffled all the sequences to randomize the backgrounds, and then we reimplanted the positive Homer motifs into their original positions in each positive and negative sequence. For negative sequences specifically, we randomly chose a shuffled positive sequence as a background to ensure GC content matching between positive and negative sequences. The advantages of this approach are that any motifs that weren't discovered by motif discovery are erased by dinucleotide shuffling, so we have some notion of ground truth features. Additionally, real motifs at their original positions model complex cooperative TF binding patterns. In this work, we generated our positive set based on 400 base pair regions accessible in one of the 5 listed cell types. As our negative set, we used an equally sized set of regions that are not accessible in the cell type of interest, but are accessible in other cell types. On the right, we have a heat map illustrating the motif co-occurrence patterns for the A549 dataset. Red indicates the motifs are more likely to co-occur relative to a null, where the motifs are randomly distributed. This heat map clearly illustrates that the motifs are not randomly distributed across sequences. Our simulated dataset also maintains positional patterns, such as some motifs being more likely to occur towards the center of a sequence as opposed to the flanks. The histogram shows that positive sequences are more likely to contain instances of a particular motif near the center of the 400 base pair region compared to negative sequences. As an aside, for those familiar with the GradCam interpretation method, note that the presence of such positional patterns indicates that we should not prefer to use GradCam, which discards positional information and thus cannot reflect the fact that motifs can have differing importance depending on where in the sequence they occur. Coming back to the simulation, one disadvantage of our approach is that we are limited by the motif discovery method, since we could have missed motifs or identified false positives. We pre-initialized our model with the deep sea beluga and basset weights and fine-tuned on our simulated data. All models achieved AUROCs and AUPRCs in the range 0.7 to 0.8. To briefly go over popular interpretation methods that we compared against in this work, ISM is a popular perturbation-based approach, while deep lift, integrated gradients, and grad times input are popular backpropagation-based approaches. The deep lift and integrated gradients methods rely on choosing a reference or baseline input. In the poster, we have some guidelines for how to pick a good reference. The choices we consider in this work are all zeros, average of 1 hot encoded inputs, and multiple references generated by dinucleotide shuffling the original sequence. To calculate AUPRC for motif recovery, we follow the method of Ku et al. For a given cell line and interpretation method, which computes the average AUPRC across all accessible sequences for distinguishing positive positions from negative positions, or motif regions from non-motif regions, based on absolute value of sequence importance scores for the interpretation method. In our Motif Recovery AUPRC comparison of different interpretation methods, we find that deep lift and ISM tend to perform best and second best, and grad times input tends to perform more poorly. Also note that integrated gradients sometimes performs more poorly than grad times input, which we will discuss later. When comparing different deep lift references, we find that shuffled reference works better than zero or average GC reference for deep lift. Also, deep lift performance initially increases with the number of shuffled references before experiencing diminishing returns at around 20 shuffled references. We also find that integrated gradients with 20 interpolation steps and 10 interpolation steps gives very similar performance, whether using 10 or 20 shuffled references. As we saw earlier, integrated gradients sometimes performs more poorly than even grad times input. We speculate that this is due to the linear interpolation that is done between the reference input and the actual input. Looking at the histograms of sigmoid logits during interpolation between original and shuffled A549 sequences, we see that the output on the interpolated inputs sometimes goes outside the distribution of the output on real inputs, which could be related to the poor performance. Here's a visualization of the scores on a sequence where integrated gradients perform poorly. The ground truth motif is highlighted in blue. One direction for future work is to compare our results with the 2019 work by Anupama Jha and colleagues, which proposed more sophisticated choices for the interpolation path for integrated gradients. In summary, we present that simulations guided by real data are a promising approach for benchmarking interpretation methods. Using our simulation, we uncovered several findings about how best to select hyperparameters, such as the choice of reference or the number of interpolation steps. We anticipate that our work will be a useful starting point for improving interpretation methods for regulatory genomic data.